Translation is the process of making a protein via a polypeptide chain of amino acids. There are 20 amino acids that can be used to build a protein, and each one is specific to what is translated from a 3-nucleotide codon in the mRNA. The process of translation takes place in the ribosome. This video is specifically focused on translation in eukaryotes. Therefore, unless otherwise noted, the term eukaryotic ribosome refers to a ribosome in the cytosol. To better understand each step of translation, we are going to start by looking at the composition of a ribosome. Next, we will explain how a tRNA molecule obtains and adds specific amino acids required to build a specific polypeptide chain. And finally, how the ribosome manipulates those tRNAs according to the mRNA transcript to translate and ultimately make a functional protein.
Ribosomes are formed from the assembly of many different proteins in rRNA. The synthesis of eukaryotic rRNA happens in the nucleus, and synthesis of ribosomal proteins happen in the cytosol. Final assembly of these constituents into a ribosome happens in the nucleolus. Each ribosome is composed of structures called the large and small subunits. In eukaryotes, the small subunit is termed 40S, and the large subunit is termed 60S. The 60S subunit contains 3 discrete sites that act as binding zones for tRNA during specific points of translation. These sites include the A, P, and E sites. Together, the 40S and 60S form an 80S ribosome. The designation S refers to the rate at which these subunits sediment when subject to a centrifugal force.
During translation in eukaryotes, the mRNA lies on the surface of the 40S subunit between the 40S and 60S subunits. In order for the mRNA to initially be recognized by the ribosome, the mRNA is first recognized by the initiation factor EIF4, which is a multi-protein complex that recognizes the 7-methylguanosine cap at the 5' end of the mRNA. Initiation factor EIF4 then facilitates the binding of the 5' end of the mRNA to the 40S ribosomal subunit. The 40S ribosomal subunit reads the mRNA strand 5' to 3' and looks for a start codon to begin translation.
The correct start codon is flanked between a very specific base sequence known as the Kozak sequence. When the 40S complex reads GCC, GCC prior to the start codon, and C after the start codon, the complex knows to begin translation. The initiator tRNA in eukaryotes carries methionine, which codes for the start codon AUG on the mRNA. A eukaryotic initiation factor, EIF2, binds directly to the initiator tRNA carrying methionine, and EIF2 recruited to the 40S subunit in the P site.
Prior to the initiator tRNA binding to an EIF2, an uncharged tRNA becomes charged with the specific amino acid methionine. The tRNA is charged by an enzyme known as amino-acyl tRNA synthetase, which catalyzes the attachment of amino acids to tRNA molecules. Each amino-acyl tRNA synthetase is named for the specific amino acid it attaches to the tRNA. For example, the tRNA coding for the start codon in our mRNA strand receives its specific amino acid methionine from methionine tRNA synthetase. Once the initiator tRNA is charged, EIF2 recruits it to the P site to begin translation. Once again, translation of the mRNA involves binding of a 3-nucleotide sequence on the mRNA, designated as the codon, to a 3-nucleotide sequence on the complementary or isoacceptor tRNA, designated as the anticodon. Codons are important because each binding tRNA carries a specific amino acid essential for the synthesis of a specific functional protein. Amino-acyl tRNA synthetase continues to catalyze amino acids to the tRNAs following the initiator tRNA. However, these tRNAs are recruited to the ribosome in the A site by an elongation factor, EFTU. Elongation factors EF, EFTS helps EFTU function correctly. As the ribosome continues to the next codon on the mRNA, another elongation factor known as the EFG moves this uncharged tRNA to the E site and a new charged tRNA into the P site. Another charged tRNA will enter the A site at this time. This process is known as translocation. As the tRNA moves from the A site to the P site, the polypeptide chain is transferred to the P site. Once a tRNA is bound to the P site, its peptide chain is then catalyzed and transferred back to the A site by peptidyltransferase. The red arrow we include in this segment helps organize the movement of tRNAs through the elongation process of 1. Becoming charged, 2. Recruitment to the A site by the EFTU-EFTS complex, 3. Catalyzation of the polypeptide chain in the A site, 4. Translocation of the tRNAs, and 5. Repeat. As the polypeptide chain grows in length, the tail exits the ribosome through a channel. This channel is located within the P site of the 60S subunit. Since peptidyltransferase is no longer needed at this point, we removed it to better show the elongated polypeptide chain. The final stage of translation, known as termination, occurs when a stop codon is reached in the mRNA. In this video, the stop codon is UAG. The stop codons are not recognized by a tRNA with a complementary sequence. Therefore, an amino acid is not added at this point. Instead of a tRNA, stop codons are recognized by proteins known as release factors. Release factor, known as ERF1, is responsible for recognizing the stop codon in eukaryotic translation. Another release factor, known as ERF3, binds to the ERF1. This complex cleaves the polypeptide from the tRNA. The tRNA is released and the ribosomal subunits mRNA and release factor dissociate. If the mRNA was correctly translated and the correct amino acids were included in the correct order in the polypeptide chain, the chain will fold into a functional protein, which is the ultimate goal of translation.